bioengineering. So, our basic idea is you have cells such as bacteria and have little pumps in their membranes which allows them to move compounds in and out selectively. And we think that we can harness this and use it for a variety of applications in medicine, pharmaceuticals, or in bioengineering. So, we have here an animation of one such pump in action. As you can see, it takes in a substrate, then changes its conformation, allowing the substrate to move to the opposite side of the membrane. This substrate could be a drug that the bacteria synthesizes and pumps out into a tumor, or it could be a toxic compound that it takes up from the external and concentrates inside itself. Now, our basic idea was then to apply this to a problem, for instance, diabetes. So, looking forward, a diabetic goes to the doctor and he takes a blood sample, gives this to a scientist who then engineers the bacteria so that they're specific to that individual. They take a capsule containing a set of bacteria, which we call the sensors. These enter the small intestine and locate to the small intestine wall. They embed themselves in the wall and then grow to form a filament, which links the small intestine to the large intestine. Having done so, this then is capable of conducting an electrical current. Then they take a second capsule, which contains what we call effector bacteria, which are the ones who are going to actually work to treat the diabetes. These go to the large intestine, where they integrate themselves into the normal intestinal microbiota. So this is how the bacteria will regulate the glucose absorption. Diabetic eats a meal, it's digested in the stomach, and glucose begins to be absorbed in the small intestine into the bloodstream. And the sensor bacteria will absorb some of the glucose, metabolizing it and using the energy to send a small electrical current through the filament to the large intestine. The sensor bacteria in the large intestine then release a signaling molecule which recruits the effector bacteria so that they cross over from the large intestine into the bloodstream. Once in the bloodstream, these bacteria will synthesize insulin, and then using these little pumps in their cell membranes, pump the insulin out into the blood. The insulin then allows the diabetic to take up the glucose as they would if they were injecting it, and they'll then use this to fuel themselves as normal. Once they've absorbed sufficient glucose, the bacteria will use another set of pumps to absorb any excess glucose left in their bloodstream. They will then return to the large intestine and be excreted as usual. And a small number of bacteria will always remain in the large intestine, and these can then reproduce normally so that there's a fresh set of bacteria always present for the next time you have a meal. So we carried out a SWOT analysis, and I think the great strength of ours is it's innovative. It has wide applications across medicine and pharmaceutical, not just diabetes, but in cancer treatment, and also outside of the body in pollution remediation. Stanford University have recently carried out some studies in bacteria, using them to carry out biocomputing, and this could be integrated with our idea as a control mechanism. Looking at our costings, in the first 6 months, we used the money, hopefully from the Science Gallery, to showcase our idea at various forums, such as Ars Electronica, Grow Your Own, and then kind of get feedback from that. Then moving forward, we would like to carry out proof of concept experiments, hopefully with funding from bodies such as Science Foundation Ireland, the Irish Research Council. We'd also like to liaise with end user organisations such as Diabetes Ireland and other interested parties and other universities so that we could get feedback from them. And then moving on forward into the future, we carry out further studies in animal models and then publish our results to get feedback from the scientific community. So we've been Parvo Pump. I'm Brian McEntegart, and there's Gavin, Bennett, Neil Johnston, and Paul Dunne. And if you have any questions, we'll take them now.
Can you say a little bit about the roles of the different team members in the project and what they would be doing if you take it forward?
Sure. Well, I'm biochemistry background, so my idea was I'm kind of dealing with the pumps themselves and the mechanics of that. And Gavin, you might want to say.
Well, I study human health and disease, so I kind of looked at the diabetes and the current treatments, insulin, and maybe the disadvantages of that, and maybe how we could improve on that.
And my name is Neil Johnston. I'm not from a science background. I'm from an arts TSM course. And I've been looking at some of the ethical issues, reading up about those that might come up with this project. Also, mainly with safety issues, public relations issues relating to this kind of project.
Hi, my name is Paul Dunne. I'm from NCAD, so I was in charge of the graphics on show and trying to understand how the user would interact with the product and how we could display their behaviour towards it, and trying to make it as easy to use or easy to understand for a patient so that when they're in the hospital there's no confusion or no scare factors or anything like that.
What was the initial inspiration?
Well, I just had an idea from my background in biochemistry that these pumps, I'd read about them, I thought, well, we could use them in all kinds of ways. And then when we went into it, we kind of, diabetes, it's such a topical issue, I mean, with increasing in the first world, so we kind of thought, well, we could use it as a treatment for that, and we kind of, then with the others, we kind of moved forward and we kind of thought of the artistic, we kind of, with the animations, we kind of came up with further ideas and integrated other aspects, and they kind of just developed from there. It seemed kind of the prevalence of diabetes, and I suppose just no one else had done this either that I could see in the literature.
If you were to present this in an artistic format, in an exhibit in Ars Electronica or otherwise, how would you present the information you've given in this presentation in a standalone form?
Well, we did think of various ideas. I mean, animations seem to lend themselves well to giving an idea because it's micro scale, so animations kind of allow you to see what's going on in a way. I mean, we would think also models, I mean, actual physical models that would kind of give an idea of the processes. I mean, Paul, you kind of came up with it.
Yeah, we were like, we kind of designed out the exhibition to be in the shape of a cell, and the hook would be that the customers at the exhibition would go into the large cell, would be very large, and they go in, look around, and they kind of see all the different aspects of the cell and how it would work and how the effector bacteria and the sensor bacteria, there'd be all different kind of stations within the cell where they'd be able to go to. We're also looking at how they'd be able to recognise our exhibition from other people's. We were thinking like, so when they enter in the exhibition as a whole, they'd probably be at an exhibitions for a couple of hours, so we were thinking of like a walking stick that they could be provided to them, be in the shape of our electronic wire, like all the bacteria on it. They can just be walking around with that, and then they know that's what they got that from. They'll see it there. It's like they'll know exactly what the connection. Then they might be more interested in going to see that. So.